Okay, welcome everybody, and we're very happy to have Jocelyne Vreede here from computational chemistry of the Van het Hof Institute for Molecular Sciences here in Amsterdam, and she's going to talk about atomic interactions to cellular processes.
Okay, thank you, and nice to be giving a talk here in this deep seminar series. And yes, I will be talking about from atomic interactions to cellular processes. And okay, for those who know me, you know I'm a chemist, so that's my background. That's usually what I will fall back to. But I will use basic physics-based models to explain processes in biology. So okay, to set the stage, atoms, building blocks of matter. So this is Bohr's atomic model and then the periodic table of the elements. So basically this is how atoms look like, and this is all the building blocks that we have. And then these atoms, they have interactions between each other, and I have here a simplified description of how these what these interactions are. There are bonded interactions and also non-bonded interactions, so-called van der Waals interactions, and electrostatic interactions, which I try to indicate here with the blue and the red atom. They have different charges, so they interact. It's all very simple. This all derives from the electronic structures of atoms and how that interacts and so on. But I just use this simplification. And actually, these are simplifications that have been used already for centuries, as this slide that I took from a Nobel Prize winner in 2013. So bonded interactions you can describe basically by Hooke's law for bonds and angles, and then torsions. How to describe that was already discovered by Fourier. Then we have the van der Waals interaction and the Coulomb interaction. And as you can see, we have known these already for centuries. Okay, well, I already mentioned Hooke. Hooke was also very instrumental in discovering cells, the building blocks of life. Here you see just a few examples and a bit more detailed description that explains the difference between eukaryotes and prokaryotes. But yeah, I'm a chemist, so then I actually look at chemical reactions, and here you see all the biochemical reactions that take place in a cell. And I say all biochemical reactions, that's already really a very much an overstatement, because this is also a simplification of basically the reactions that need to take place in a cell to be alive and functional. I already said here biochemical pathways. So a biochemical pathway is basically a set of chemical reactions that are linked. So they either succeed each other. And in this way, a cell can do things. And these do things, I'm a bit vague about that, because a cell can do many things in order to stay alive. And in my seminar, I will discuss two types of cellular processes, how a cell interacts with its environment, and gene regulation. And I'll give some chemical basis for that. Okay, so to interact with an environment, so basically these are some slides that I actually took from my lectures on biochemistry in the chemistry bachelor. So here's my very simple picture of a cell, with the green indicating the membrane. And we have inside the cell and outside the cell. So very simple. So and this membrane, it poses a barrier between the inside connecting to the outside. That's all fine, very nice actually, because this way a cell can actually generate energy, store energy, and this is all very essential for life. But in order for this cell to survive, it also needs to be able to transport molecules over the membrane. And it also needs to be able to detect changes in the environment and then generate a response so that it can react to these changes in the environment. Okay, how a cell perceives and responds to change in the environment. Well, here is a list of signals that a cell can perceive. It's by no means exhaustive. It just gives you an indication of the type of signals a cell should be able to respond to. And these are both physical and chemical. And maybe even biological. If you say a hormone, it's actually a molecule. So I would say it's chemical. And these signals, they are actually detected by proteins called sensors or receptors. These can be either membrane bound or just in the cytosol of the cell. And then there's a process of signal transduction. This is a biochemical pathway, a series of chemical reactions that then ultimately lead to a response of a cell. And this response can be many things. The cell can start to move or stop moving, can start or stop various other biochemical pathways, can start or stop gene expression, or start to depolarize a neuron. And then as an example of this last thing, here's the pathway for vision. So what happens here is there, all the way here, maybe for the Zoom people can try to highlight it like this. Okay, lost my mouse, yeah. Light is detected by this protein called rhodopsin, and then rhodopsin undergoes a conformational change. Then enzymes are activated, lots of enzymes are activated, which then in the end lead to opening of certain ion channels, which in the end results in the depolarization of a neuron. Well, there's lots of amplification going on here, and there's such amplification, it's so effective that we can actually detect 10 photons as a light flash. Imagine 10 photons, and it's complete dark, and you will see it, and then the response that you will have is that you will probably turn your head towards that light flash thing. What happened there? So this is how effective such a signal transduction pathway can be. Okay, even though...
Is the physics here any important? Like this is a different bilayer people sometimes describe this stuff. Using physics, but
in this particular pathway, the bilayer is just a barrier between inside and outside of the cell. And it contains rhodopsin, so that is the molecule that detects the light. But otherwise, yeah, that becomes a very intricate.
Bilayer or the elasticity of the light, that doesn't play.
I wouldn't say in this particular process, no. And maybe in other signal transduction processes, yes. So, for example, touch, that is very sensitive. But then I'm a bit on, because I know quite a bit about light reception, not so much about mechanosensing, but you have certain mechanosensors that can detect whether a membrane is curved or not, and also temperature depends on that, because the membrane can undergo a phase transition and then becomes thicker or thinner depending on the temperature. So with thinner, it's more fluid-like, and then the membrane thins. And this also can be detected by certain proteins and then initiate a signal transduction process. But these are not the processes I want to talk about. I actually want to talk about this. And this is about plants, and that plants do not like sodium. And what you see here is two plants that grow on soil that has increased concentration of sodium. So on the top, there's no sodium in the soil. And here on the bottom, there's quite a lot of sodium in the soil. And then you should actually focus on Arabidopsis thaliana. That plant really does not like sodium. I mean, it's really happy, it grows happily there when there's no sodium. And okay, there's maybe now one pot that actually has some leaves here. Okay, maybe here to point out this plant, this Parvula plant, that's actually a halophyte. It likes sodium, so it actually thrives on sodium-rich soil. That's actually to indicate that some plants can actually handle high concentrations of sodium, but better than others. But unfortunately, most plants do not like saline soil, and in particular, food crops are very sensitive to this. And this is actually a quite problem, because climate change actually makes arable soil much more saline, because we have to irrigate more. This water then evaporates on the soil and then leaves behind some salt. So this is some context for why we actually want to look at sodium sensing in plants. Well, looking a bit more closely, what do plants do if they actually come close to more sodium-rich soil? Arabidopsis thaliana then actually has roots that grow away from the high sodium concentration soil. So here, the dark one, that has a high sodium concentration, here low sodium concentration. You see that the roots grow away from that. This is called negative halotropism and has been studied extensively. And it seems that this sodium, it sort of leaks into the root cells, and well, all this high sodium concentration affects all kinds of biochemical pathways, and then in the end leads to, well, actually it doesn't lead to the root growing away, it actually leads to the plant being very unhappy, to put it in a non-scientific way. However, so lots has been also found out about how this halotropism works and what kind of proteins are involved. The problem, however, is that it is not known how sodium actually initiates all these processes. And then we can do a very simple model of how this, what this would look like. So here again, I have my simple cell with a receptor protein and some sodium ions. Okay, and what happens if one of these sodium ions binds and the receptor does something and then a response is generated, and we know the response already, that is this negative halotropism. Okay, but now what? We want to know, what is this receptor? Okay, we can do then a bit of conjecturing. And already Darwin found out that plant seeds are affected by sodium. And also, well, much later was found out that ethylene receptors are also involved in seed germination under salt stress. And there's some some ethylene receptors that accelerate the germination, and some ethylene receptors delay the germination. Maybe for those of you who don't know, ethylene is a very simple hydrocarbon, and it's also involved in the ripening of fruit. It's a plant hormone. Okay, but these ethylene receptors, they are ubiquitous throughout the whole plant and in any plant, and they mediate a lot of different, basically they integrate a lot of signal input and then in the end give a result. 5 are known for Arabidopsis thaliana. That's what those you see here in the picture, they're all membrane bound. The domain composition of these, oh yeah, maybe good to know. So ETR1 and IN4, they accelerate the seed germination under salt stress, and ETR2 slows it down, and the other two actually don't do anything with seed germination under salt stress. How this was done is basically knocked out these receptors and then looked at whether the seeds would still have an effect on seed germination still was affected. Okay, these receptors, they have a very general domain structure. They contain a transmembrane sensor domain. The sensor domain here is ethylene binding domain. They contain a so-called GAF domain and a transmitter domain. And this is generally how signal transduction works. You have sensor domains that detect the signal, and then transmitter domains that pass it on or amplify it even. This GAF domain, okay, the GAF is an abbreviation, and it's an abbreviation of the first 3 enzymes that actually had this particular structural domain. So the name itself is not so interesting at this moment. It's just an easier way to say we found a particular structure. These GAF domains also were found to be sodium sensitive. So this is the conjecture. We actually now can try to see if this ETR1 GAF and IN4 GAF, if they are actually sodium sensitive. Okay, so how can we do that? Well, we can do it here as follows. We just zoom in on this receptor domain and zoom in on the sodium binding. But of course, this doesn't tell us anything. This animation that I made, we need much higher resolution in space and time, of course. And this we can actually get by molecular dynamics. Okay, and here my atomic fluctuations and atomic interactions come back. So what do we do in molecular dynamics? We have a certain potential and a molecular system, and then we can actually compute the time trajectory of this molecular system given a particular potential. Because we know the forces that act on the atoms, and because we know the forces, we can predict how they change position, how the particles change position. And these forces, they come actually from this these interactions I explained at the very beginning.
Can you remind us what's the
I'll do that in a moment. Yeah. First, molecular dynamics. So here we have a two-atom system. Particles have a position and a velocity. And well, they also have interactions with each other. So what happens is we have a time t, and the atoms have a position and a certain velocity. From this, we can actually get the forces, so what forces they exert on each other. From these forces, we can then predict the new positions for small time step ahead. And then we get the new forces, and so on and so on. This is why we need a supercomputer, because to do this by hand is super boring. Okay, what are these?
We evaluate the value of the force at the given position. Because the forces are given by the potential.
Yes, sure. We know the potential, but the potential then, because it has a certain position, it means that here particles are further away from each other, so they might feel a lower force. Yes. Yeah. Okay, that's what I mean. Yeah.
Do you do this deterministically, or you add stochastic fluctuation?
That depends a bit on which thermodynamic ensemble we do. But this is okay. Here I wrote the two particles, so then you can still think about deterministic and so on. But I look at many body systems, so any small, really tiny variation at the beginning will just lead to huge differences, larger differences later on. So if I want to simulate at constant temperature, that means I have to couple it somehow to a heat bath, and this I can do by introducing some stochasticity in it. Yeah. Okay, then to come back to your question, so what is this, where do these forces come from? Then is again this slide. So we have bonded interactions. So basically two atoms, they form a bond, three atoms, they can have a bond angle and then still interact with each other. Four atoms, they can rotate along a certain bond. So you see here. Non-bonded interactions, these are the van der Waals interactions. So basically, fluctuations in the electron cloud of an atom can that can result in an instantaneous dipole for that atom, and that can induce other dipoles in surrounding atoms, and this leads to an attractive interaction. Also, atoms cannot sit on top of each other. That's a repulsive interaction. That's all summed up basically here. And then we have electrostatic interactions. I think that answers your question, right? Yeah, that's fine. Okay, yeah, so in the type of molecular dynamics simulations that we do in my group, we don't evaluate where the electrons are. We just say, okay, there are bonds and there are atoms that are not bonded. So before we actually set all of that. Okay, so how can we then, yeah.
So for all these, this is all classical. Is quantum physics ever important for any of these processes, or is it completely unimportant and you can always ignore it? Like the photon absorption by the
there you need quantum chemical description. Yeah, you cannot get away with.
Most of the cell dynamics need to be completely classical, or?
No, because as soon as a chemical reaction happens, you need a quantum chemical description, because then bonds are being made and broken. So as soon as even for a proton transfer, well, there are some methods where you can have sort of a simulation at constant pH, and then you can say you have some sort of dummy proton that is, depending on the electrostatic environment, it's slowly switched on or off or something like that. But even proton transfer reactions should be modeled quantum chemically.
Right. These processes don't involve.
No, I had a certain bias in selecting. Yeah, in the work that I do, I have a certain bias to look at processes that you can model with classical mechanics. Yeah, definitely. But I'm not saying that you don't need quantum chemistry. It's just that I don't do that. Okay, so what is the workflow? So we have a sodium sensor candidate. Then we do all atom molecular dynamics with sodium. You see, okay, this is just really an illustration of the workflow. This is actually not really the protein that we're looking at. So what we did is we did 5 times 1 microsecond all atom MD simulations and analyzed that. And from that, we can then get predictions on where the sodium binds and if there are any conformational changes. And yeah, I usually use this as an example because you do see a conformational change here. So this is basically prediction of sodium binding sites. And as a control, we do the same with potassium. And well, it's not actually the potassium data that you see here, just to illustrate.
Will you explain the difference at the level of the model of doing one or the other?
Yeah, okay, but I will show some results in a moment. Yeah, it's just basically as a control, because of course I can do all these simulations with sodium, but then if potassium does exactly the same thing, you could say it's not really a sodium
No,
sensitive.
I just meant what is the difference at the level of potassium?
Oh, a potassium has a bigger radius. So it's a bigger molecule, and also the non-bonded interaction parameters are different. This is actually quite tricky because we had to optimize the interaction parameters for proteins with sodium and with potassium to get this right. There was a lot of work that I'm now sort of sweeping under the carpet and not mentioning, but
so you actually have some kind of understanding of what the parameters of the interactions are. You can get them from some.
So, okay. I go back to this slide. So the interaction parameters that are relevant for ions is just a single particle, and we assume that the charge is just a single point charge on the ion. And the ion still has a radius, which is relevant here for this interaction parameter. It has an R0 basically and an epsilon. And this epsilon and R0, that depends on the atoms that it interacts with. And yes, these have been optimized actually for, okay, I had a collaboration actually with someone who really looked at halophilic proteins and optimized all these interactions of proteins with sodium and with potassium. But I thought I want to show a lot, so I thought I'd just skip this a bit. Okay, so as analysis, we actually just counted the number of snaps for each residue in the protein, which is given here as on the horizontal axis. We then count how often we find a sodium or potassium ion within 0.35 nanometer of that residue, and then actually from that you can calculate the probability. And what you see is that sodium really likes to be much closer to the protein than potassium. And as I said, well, we carefully optimized all these interaction parameters. So this is not an artifact. This is actually, so yeah, potassium has a different hydration shell, just likes to be less around proteins than sodium. So we also then identified regions, so where does this sodium then bind? And also what we did is look at, so here we look at for each residue whether we find an ion close or not, and that's basically what we define as proximity. Here we looked at how what is the probability of finding two residues close together, and that's not even that, it's it. And then we did that for sodium and for potassium, and then subtracted the contact map for potassium from sodium, so that we actually can really focus on the differences. So are there different contacts for sodium? If so, then they are highlighted in red here in this map. And if they are contacts that are only there when potassium is present, then they are highlighted in blue. Which actually then allowed us to identify that this region, that's basically around here, there something happens. So the different connections, different contacts made when sodium is there than when potassium is there. And based on that, we were then able to actually select a few mutations. So based on these results, then some mutants were made, and these mutants were actually introduced in the plants. And then I have to actually end this story on a cliffhanger, because these mutant plants are still growing. So we cannot say yet whether our prediction turned out to be valid. But this was a nice, I think at least a nice introduction into molecular dynamics and how we can actually already gain quite a bit of information on cellular processes by just doing this quite simple MD. So the prediction that we made is that certain amino acids in the protein, in this ETR1, are involved in sodium sensing, and that sodium actually binds close to these residues. And it does not do that with potassium. And then when sodium is not there, the protein has a slightly different shape. So the prediction is that if you mutate these residues to something that cannot no longer bind sodium, so the residues were negatively charged, we mutated them to alanine, which just has a methyl group, then we suspect that the plants will not have this negative halotropic result.
So here you're growing this stuff with a lot of sodium to see.
No, actually not. So I'm not a plant biologist, so now I have. So how this works is that you have to introduce somehow the mutation in the plant, and the plant has to grow seeds. Then these seeds have to grow out to plants and then produce again seeds, and then those seeds, then we can test for this negative halotropic response.
So this is the first stage?
The second stage, and they have to produce seeds now, and then we can check. Also, well, if you grow them on soil that doesn't have any sodium, I think they would still be viable. Yeah.
Do you expect them to grow better in soft soil?
Actually, we would just wanted to look at this negative halotropic response, because that is very specific. And if we have now removed this particular interaction and then it no longer exhibits this response towards sodium-rich soil, then we have actually found the molecular center. Yeah, it is a very tiny detail, but so growth rates and so of those plants, I don't think I should say anything about that. So, okay, more questions? But yeah.
Do I understand it right that you already know exactly the original structure of the protein? Or is there also uncertainty about that structure?
So first we did some homology. So the structure of this GAF domain is not known. Okay, the other structures of GAF domains known, but not of the ethylene receptors. So we first used homology modeling. That turned out to be not such a good prediction, but we then used AlphaFold. I don't know if you've heard of AlphaFold. So this is a protein structure predictor based on neural networks, and that actually works like a charm. The protein is very, very stable, as in the structure that it predicts looks like a GAF domain, but the interactions are such that when I put it in water in the simulation, it just stays together. So I would say that's our basis.
Is your work also evaluating the prediction that AlphaFold 2 that you see?
Yes.
Or is that
I'm
already very confident that the prediction is correct?
Well, based on sequence alignments, we identified a region as a GAF domain. For this, we know what the general structural features are. So we could basically at first say, okay, we take some other GAF domain and use that as a template. That didn't work so well, but with AlphaFold, it seems that the core of the protein, that seems to be well packed, such that if I put it in water, it doesn't fall apart. However, you make actually a good point, so we should actually check this, and we're actually also doing that using infrared spectroscopy to look at, and also CD spectroscopy to look at the structural content of these proteins. But purifying these GAF domains is also a bit tricky and actually requires someone with good biochemical skills. So yeah, okay, this project, I ended really on a cliffhanger, so still lots of things are going on. And I think the simulations have here played an instrumental role in understanding more about how this sodium sensing in plants work. And this is also a good moment to acknowledge the people that worked on this part of the project. So Guido Giannetti was an exchange student who worked in my group and did all the simulations. Tom van der Meer is a plant biologist who also did some simulations because in COVID times he couldn't do much lab work, so he thought, let's try this. And he did a lot of the initial stuff, and actually then a lot of things went wrong, which Guido could profit from. Christa Testering is the head of the plant physiology group. And with her I talked a lot about how to actually approach this whole problem. And Giulia Gibertoni, she is involved in doing some measurements. Tom also tried to purify these GAF domains, but there are lots of issues with that. Also, to actually get pure samples is really tricky. So it turns out that sometimes with these samples, then some protease was also in the sample, which meant that the protease was actually cutting the actual protein to pieces, so we couldn't measure anything. So, so then simulations seem so much more clean and if they work. Okay. The other thing I wanted to talk about was gene regulation. And this is maybe, I mean, I'm very interested in this, but I'm not sure if I can already simulate this. Okay, gene regulation, that has everything to do with transcription. And to remind you, transcription, what it is again. So we have the DNA in blue here, and then RNA polymerase, that's the purple blob, that binds to the DNA and then opens it up and then starts synthesizing RNA by basically copying what's on the DNA. I think all very familiar to you. So here is my simplified representation of this. And to regulate the transcription of a gene, that is mediated by so-called transcription factors. And this is what we can also call direct regulation. And these transcription factors, they bind on to close to sites or on sites where RNA polymerase starts to make DNA. How this works, RNA polymerase binds to a specific site and then starts making the RNA. This is also usually very specific. And these transcription factors can either enhance or the affinity for of the RNA polymerase for that particular transcription site, or actually just bind on top of the transcription site and completely block it. So that's the direct regulation. Well, this is an example where the transcription factor actually enhances transcription. This RNA polymerase binds and starts making RNA. But this is a very simple representation. This blue line here is not all of the DNA. It is part of a whole genome, which is much, much larger, which I now indicated by this lighter blue line. And then we can also have indirect transcription regulation, because somehow the conformation of the DNA could be mediated even by proteins, can be such that, so what you see here is actually that the DNA has formed some sort of loop, and transcription is blocked because the RNA polymerase can no longer read the gene, because there's just basically a roadblock there. And this is what we call then indirect regulation. So the accessibility to the gene is mediated by so-called DNA organizing proteins. And one of the most famous ones is chromatin. And you might think, well, what a simplistic structure. Well, this is actually what some DNA organizing proteins do. So what you see here is atomic force microscopy images of a plasmid. Here is just a plasmid on a mica surface. And well, DNA is negatively charged, so this tries to approximate a circle, still fluctuations. And here is the DNA with, well, let's call the protein HNS. Don't have to know what the actual name stands for, but this HNS actually forms bridges between two DNA strands and brings it together. So this actually can form a roadblock and then prevent gene transcription. And that's actually also what HNS, its function is. Okay, so what is then the structure of such an HNS DNA complex? How does it form? Well, what I've made here is just some cut and paste in PowerPoint, so not based on anything physical. Because again, finding the answers requires, again, resolution at high resolution in space and time. So again, molecular dynamics simulations. And this system is much, much larger than these sodium sensors I was looking at before. So now the numerical aspect of molecular dynamics becomes important. So let's say I have long stretches of DNA and a couple of these HNS molecules, and I just throw it together and wait until it forms. Let's say it takes about a millisecond for such a complex to form. Well, the time step that I use in molecular dynamics is 2 femtoseconds. And let's assume that for such a big system, one time step takes 1 millisecond on a supercomputer. Oh, sorry, wrong arrow. Yeah, then I would have to wait way too long. Then I don't want that. Seems obvious, right? So what can we do to overcome this problem? Well, one way that we can do is basically lump groups of atoms together and then describe them by bigger particles with effective potentials between these particles. And this is one way. We can then have one binding site is 1 particle and the protein is 3 particles. And a while ago, I developed such a model. Okay, this is quite a detailed slide. I didn't adjust it for this presentation. But what we wanted to simulate is these AFM pictures that I showed before. And then instead of having every atom of each nucleotide there, we now have this plasmid, which sums up to 179 beats in this model, and the protein is approximated by 3 particles. And then we have a interaction potential, which consists of the potential of particles of DNA interacting with each other, protein particles, intraprotein interaction, interprotein interactions between the protein and DNA, and some sort of confinement potential.
Is there a possible way of deriving these effective interactions from the more microscopic ones, like the ones you just said?
Yeah, I'm just going to show that.
Okay.
Yes, so in order to know, to say something about the interactions between the protein and DNA, I first have to know a bit more about the protein. So a lot of these other potentials were already derived before and were indeed based on more microscopic simulations or all atom simulations. Yeah, and that's indeed actually the rest of this seminar. Okay, interaction between protein and DNA. Well, with this interaction potential that we have, we can actually say the protein really likes to sit on DNA, which is shown here. This is basically just a 2D plane. And the darker blue, the more attractive the potential. So these blue beads really want to sit close to the DNA. And on the other side, if another DNA particles approximate the protein, you see basically the same. But then for this intra-protein interactions, well, we model the protein by 3 beads. So we have bonded interaction between the 2 particles and a bending potential between the 3 particles. But this bonding potential, that's basically just a stretch. And we know the equilibrium distance more or less because some crystal structure data is available. We don't know anything about this bend, so we had to approximate that. So we made a flexible potential and a stiff potential. And then actually ran simulations of that. So this is with the flexible protein bending potential, and here with the stiff protein bending potential. You can already see the differences. So in red is the DNA, and green and blue is the protein, and with the stiff potential, the DNA is much more compact. Well, if we want to compare this to the AFM results, and say that the flexible potential is actually closer to what you get in experiments. I know this is all very hand-wavy, but otherwise I won't have time. Because, well, this was still a very simple model, and now we want to actually tune all these interactions within the protein, so this bending potential. I mean, I just said flexible and stiff. It basically meant that I have set some parameter to either very high value or a low value. The interaction between proteins that can be tuned, and also specific binding sites on the DNA. And that is the last bit I want to show. How do we get actually these effective potentials? But it's one thing we want to do, but actually knowing more about protein DNA complexes, that is relevant for lots of cellular processes. I focused here on transcription, but of course also in DNA replication. This is very important in DNA organization. And we want to know how do proteins achieve selective binding? Why do they bind to one particular nucleotide sequence and not to another? Well, let's go back to what is the structure of DNA. This is double-stranded DNA with the backbone. So here you have a phosphate and a sugar group, and then attached to the sugar group is the base. We have 4 types of bases. These bases form hydrogen bonds with each other. So a guanine with cytosine, adenine with thymine. Also in the orientation of these bases, so here is the sugar group attached to here this wiggly line, and here as well. These are not 180 degrees, there is an angle to it, which means that this side is slightly smaller than that side. And this gives rise that if you stack all these bases, which is often seen in depictions of DNA, you then have a major groove and a minor groove. Also has to do with the optimal overlap of these bases when they are on top of each other. Well, they still have electronic structure that needs to be optimized. So that gives rise to this well, really beautiful shape of DNA, the double helix. Okay, so what are the interactions that are important in protein and DNA interactions? Electrostatics. DNA is negatively charged, and the DNA binding proteins are usually positively charged. So you have this very strong electrostatic interaction. Hydrogen bonds, well, the hydrogen bonds, of course, between the bases, but also hydrogen bonds between the bases and proteins can play a role. And of course, steric considerations. So the DNA, depending on the nucleotide sequence, can have different shapes. And these major and minor grooves, the width of those grooves very much depends on the nucleotide sequence. Okay, as an example, go back to this HNS protein that likes to bind to AT-rich DNA in the minor groove. This has been shown experimentally with lots of different experiments. And involved in this binding to the in the minor groove is a specific amino acid motif. So I tried to see if I can also predict, if I get the same result out of when doing ND simulations. So what I have here is a HNS DNA binding domain to keep things small, and a AT-rich sequence. Here you see the nucleotide sequence, and then I did molecular dynamics. And what happens is, well, the protein moves around the DNA, and if we wait for a bit, then actually we see insertion of this particular binding motif. It really goes into the minor groove. I thought this is maybe one of the first movies that I show. Okay, but this was an AT-rich sequence. And now the question is, how does HNS achieve this selective binding? Because from this we can then derive effective potentials. Now I have a whole thing about chemical equilibrium. Just go over this fast. So we can describe this that we have a protein DNA complex and the free DNA and the free protein. And then we can say the speed of formation of the protein DNA complex, that depends on concentration-free protein and concentration-free DNA and some rate constant that we call K-on. For the dissociation process, that depends on the concentration of protein DNA complex and the K-off. And then we can say in equilibrium, then we have the situation where these conversion speeds are the same. Then we can rewrite this. Okay, I just go over this fast. This is very, chemical kinetics. So we can rewrite this equation. Then we have the concentration on one side, the rate from the other side, and that is then equal to the equilibrium constant. And the equilibrium constant says at which concentrations of the individual reactants and products you have actually achieved chemical equilibrium. And this, of course, depends very much on the type of protein and the sequence, nucleotide sequence that we look at. This equilibrium constant we can actually associate with the Gibbs free energy change of a chemical reaction, of which I have given a formula here. This Gibbs free energy change is always with respect to some standard Gibbs free energy plus basically the ratio of products and reactants. Well, at equilibrium, this Gibbs free energy change is equal to 0, so that gives us a way to find this reference value, and also a way to find the actual value of the equilibrium constant. Okay, but we actually want to get this from simulations, and then we have to count conformations, because that's just what we can do. We cannot just measure some Gibbs energy change. So what we do is, if we count conformations, we can count how many times in the simulation we have actually the complex and how many times we have the free states. And in order to go from one to the other, we have to pass through a transition state, which is the least populated one. And then the Gibbs free energy change is basically the negative log of this count. And then we get a thing like this. So, okay, here I just plotted 3 points. Here I made a more continuous line. I think you get the idea. And this is in the end what we want. We have here the reaction progress on the horizontal axis, here the free energy, and we actually want to predict somehow this Gibbs free energy change for association, if the protein binds to DNA. And then our prediction is actually that if we have high affinity, we have a certain Gibbs free energy change. This is positive because this is the favored state. We can basically already say that based on electrostatics, because only because of simple electrostatics, the protein will always bind to DNA, but we actually want to know how high this difference is for a high affinity sequence and a nucleotide sequence that has a much lower, for which the protein has a much lower affinity. So then we also think that this dissociation for Gibbs free energy is much lower. Okay, so with this procedure in mind, we can then do long MD simulations. Here is some, basically a contact count that we do. So we count the number of contacts between the protein and DNA. This is the whole derivation of it, but this is basically what we look at. And here is plotted for, in blue, is plotted the contact count for the high affinity sequence in the protein DNA complex, and in orange the low affinity sequence. And what we see is actually, even after a cumulative simulation time of 3.5 microseconds, there is no dissociation. So we can count all we want, but we just are stuck in one minimum. So, but we actually need to transition to the other minimum. Okay, how can we achieve that? Well, we can introduce an extra potential in the system. We can basically pull off the protein from the DNA. And how can we achieve that? Well, we can pull the protein off the DNA at constant velocity. That basically means we have a certain pulling coordinate lambda, which depends on t, and this changes also with t, with constant velocity. And that basically gives them, well, we have here, we also start at this specific initial configuration. So with this pulling, if we have this pulling potential, we can also measure the force, taking the derivative of this pulling potential with respect to the actual value of the system along this pulling potential. And then if we have the force, we can then also measure the work. This work is then basically integrating the force over time. Then we get the work that is put into the system, extra work that is put into the system in order to pull off the DNA, the protein from the DNA. And then we use Jarzynski's equality. That's very beautiful. If we do this, repeat this a number of times, pulling the protein off the DNA, and then average the work for each, average the work over all these separate ones, we then actually get the free energy. Well, very nice, you think, and well, this is not new. I mean, Jarzynski thought of this in 1997, so not claiming that I thought of this. I thought it would be just an interesting method, but there was actually lots to tune. So we have to pull the DNA off with a certain force constant. So we have to, you could actually explain it with walking a dog. So walking a dog is that I would be lambda t, and the dog would be my system, and the leash, the tightness of the leash, that would be this k. the speed with which I walk, that would be v, the velocity. And these need to be carefully tuned. Actually, we found there's quite a large range over which this holds. If you pull too fast, what happens is that I basically will kill the dog because the leash will just do this to it. If I let the leash too loose, then the dog will stray everywhere and will actually not follow my coordinate. But most importantly, how do I attach the leash to the dog? And well, here you think, okay, it's very simple. You have the center of mass of the DNA, center of mass of the protein, and you just extend this distance. Well, no, that turned out to be very problematic. We actually had to come up with quite a elaborate contact map parameter. In the end, I don't have time to really explain this, but with this elaborate contact map parameter, we could then reproducibly find a free energy difference for the high affinity sequence and the lower affinity sequence. And actually in a simulation time that you can run this in let's say 2 days, which is much faster than okay, there's lots of experimental data actually. So there were these huge microarray experiments where they identified to which nucleotide sequence, let's see, basically this is a summary of that. The preferred sequence to which HNS likes to bind was identified. Other things is also a combination of AFM pictures and the different types of plasmids show that HNS really binds to high affinity sequences and not so much to these lower affinity sequences. Also known minor groove binders were done in competitive experiments where you have both the known minor groove binder and HNS put together. And if you first put HNS, then the known minor groove binder could no longer bind to DNA. But it's a good question because, well, experimental validation is still underway. Okay, I have to go through all of this. Because we're actually checking the actual values with fluorescent DNA and fluorescence of changes when HNS binds to it. Here, however, it's quite problematic again to get pure samples of these HNS DNA binding domains, so we're actually now stuck at that level. Another is a collaboration I have with people in Zurich who look at chromocenters in fruit flies, and these chromocenters, they are formed by a particular protein that has 11 binding sites. And for these 11 binding sites, they have identified with basically they truncated the protein to take out more and more of these binding sites, and then could see how effectively these chromocenters were formed. And that seems to match nicely with the predictions that they did using this particular algorithm. And together with people in Utrecht, at Utrecht University, they're actually looking at this experimentally. By okay, that's so they basically look at the products of genes of transcription and how this is modulated by HNS. Yeah, so this was also the summary. So we effectively made an algorithm to a simulation protocol, actually, I should say, to simulate the dissociation of proteins from DNA. So validation and more predictions of this simulation protocol are ongoing. And as outlook, well, from these free energy differences here, we can then develop effective potential. How and what exactly, I would say that's a topic for another talk in a year or so. Okay, here I would like to take one moment to thank Thor, my PhD student who developed all of this, and I had lots of discussions with Peter Bolhuis on this. Well, to summarize, from atomic fluctuation interactions to cellular processes, I showed an example of how to interact with the environment. and showed some work we've done on understanding gene regulation. But actually what I do is I look at positions of particles and try to link that to what happens in experiments. And with that, I would like to thank you for your attention.
Thanks a lot. Very interesting talk. I'm looking at the Zoom. Maybe there's some questions.
I see David and Maurizio. They have seen this before.
They know all about this stuff.
Oh, they've seen it before in various stages.
Good, good. There's some more questions here.
Yeah. So maybe one very generic question is, I know a little bit better the AlphaFold work where claim is actually that the big advance is from not trying to sort of model the details at the molecular level and minimization to actually use machine learning and the advantage of all these examples of known substructures that very different. So is there something equivalent in your field where you can sort of take advantage of the exemplars? Or, how does it work out?
I think
or
I
do you
understand.
have to go back to the individual forces between the molecules?
I really need that, but okay. So about AlphaFold, it's very nice that exists, because that gives a lot of opportunities for starting structures with which I can do these MD simulations. I want to make a remark here that AlphaFold does predict structure, but the neural networks are trained on structures from the protein data bank, so X-ray structures, NMR structures, at certain conditions. So I don't think AlphaFold is able yet to, for example, look at the difference in conformation when you have different ions present, or when a small molecule binds to the protein, and so on. And also AlphaFold can only predict, I think, if a sequence is sort of similar. If it's completely widely different to anything that is known in the PDB, I've actually seen that in AlphaFold, and you get really weird results. So, okay, having said that, your question is, do we with molecular dynamic simulations have something similar? Well, we would love to, because that would say, well, that's the example I showed, that if you just do plain MD, for small systems you can still get reasonable predictions, but for larger systems, then you have to wait thousands of years. That is, of course, not an option. So what I showed is one example that we try to speed up things by introducing extra forces. There's another way that our group is also famous for, is by speeding up simulations by sampling only specific transitions. But up to now, it really depends on the, I would say, chemical intuition of the researcher doing this. And there's a really active line of research trying to use machine learning based potentials to speed this up. But yeah, then you would have to ask one of my colleagues for the details. So Bernd Ensing or Peter Bolhuis, they can tell you much more about this. I'm actually just waiting for them to give some ideas, because I'm usually more at the application side. I think, oh, that's a beautiful method. Let's try it on something bigger, larger, more biological.
Yeah, very young is a question, because I don't know so much about this, but is it just very difficult to know what exactly is the protein structure of the thing you want? Like, it's difficult. That's why you have to use this.
Yeah,
even for a plant, that you know what the plant is and everything, you could try to measure this. Why is it so complicated?
Yeah, because you can get stuck in local free energy, local energy minima. So, okay, let's say we start out with a random amino acid sequence. So it's a random chain with all the side chains of the amino acids there. And in order for that to fold, Well, if we want to do that on the computer, you could say, well, we can try all the different orientations that these amino acids can have. If you take for each evaluation, so let's say you have 100 amino acids, and each amino acid has 3 rotatable conformations. So that's, I have a slide on this somewhere. Well, this gives basically, we have a lot of options then to check. If you then assume that to check each option takes a microsecond, then you're still busy for a long time.
But I just meant you have the actual plant. Why can't we just measure it and see the?
Oh, that is very difficult.
Yeah, that's
you need very pure protein. So the problems I was highlighting here, that experimental conformation is very difficult. But you need to have pure set protein. That is just there's several ways to get pure protein. So you can put the gene in a bacterium, E. coli, for example, and then get it to overexpression. If you already have that, then great, because then you can then have bacteria with lots of this protein. Then you have to get the protein out of the bacterium, and actually in such a way that you only have that particular protein and nothing else. Which means you have to actually attach all kinds of tags to this protein and then run it on over some sort of affinity column so that the protein sticks to the column and the rest flushes away. But adding these tags, these may also influence the conformation of the protein, so you have to cut it off at some point and then purify that. It's actually developing a purification protocol is not that straightforward. And that's me saying I'm really clumsy in the lab, so maybe it has become a bit more straightforward, but yeah. Okay.
Do you have more questions? Yes, thank you, Jocelyne.
Okay, thank you.